In introducing the muscular system, we first have to remember that muscles are considered organs because they're composed of muscle tissue as well as epithelial, connective, and nervous tissue. And we have somewhere between 600 to 800 muscles collectively within our muscular system. There are three main classifications of muscle tissue, and this is just a recap from when we learned about tissues in Chapter 4. And the first one is skeletal muscle. And skeletal muscle is going to be voluntary, and it is going to have a striated appearance based on the sarcomeres and the Z-lines. And another feature is that skeletal muscle is multinucleated. Cardiac muscle is going to be involuntary, and it too appears to be striated because of the Z-lines, and it also has either one or a maximum of 2 nuclei per individual muscle unit, and we also have intercalcated discs that help to unite all of the fibers so that we have unison when contraction occurs. And lastly, we have smooth muscle, which is involuntary, and it is not striated, meaning that it lacks the Z-lines. And these tend to have a fusiform spindle shape where it's thickest in the middle and then it tapers off in the ends. All muscle tissue are composed of muscle cells. And so all muscle cells have certain common properties, and the first is excitability. And this is the same thing as how responsive a muscle cell is. So when a muscle cell is stimulated by the nervous system, by stretching, or another environmental stimulus, then it responds by initiating electrical changes that go across the entire plasma membrane of the muscle cells and then have internal events such as contraction of actin and myosin and the shortening of the sarcomere that equates to muscle contraction. The next property is contractility, where we have tension of a muscle cell that is generated. And so this is the same thing as contraction, where this causes the cells to shorten. The shortening of the muscle cells results in either a pull on the bones of the skeleton or a specific movement of a certain body part. The next property is elasticity, and this is when a muscle has the ability to return back to its original length when tension is released or with extension. And lastly, extensibility. So this is the ability for a muscle cell to extend in length in response to contraction of opposing muscle cells. And so, for instance, when you flex your elbow joint, you contract your biceps brachii on the anterior surface of your body. However, your triceps brachii are extending on the posterior side of your arm. And so all muscles go through this, where they undergo cycles of contraction and extension. Functions of muscle tissue include body movement, and this is where we have bones of the skeleton move when muscles contract and pull on the tendons which attach the muscles to the bone. And so this can produce highly coordinated movements like running, or simple and localized movements such as underlining a word in a book. The next is to maintain posture, and we have muscles that stabilize joints and help maintain the body's posture or position. Postural muscles contract continuously throughout the day to prevent us from collapsing. Another function is to generate heat, where energy is required for muscle tissue contraction in the form of ATP, and heat is always produced as a waste product for using the ATP. Most of the heat maintains our normal body temperature, and that's why when we exercise we tend to feel warmer, and however, when you shiver, when you're cold, because your muscles are contracting and relaxing to produce heat. Another function is for storage and movement of materials into and out of the body, where we have sphincters, which are circular muscle bands that contract at certain openings or orifices of the gastrointestinal and urinary tracts. And these may be voluntarily closed to help store material within an organ, or open to help move material out of the body. And the last function is for support, where with skeletal muscle, we have, say within our abdominal cavity, flat sheets or layers of muscle that help to keep the abdominal organs in place. And so that's one of the main functions actually of our abdominal muscles, is to help keep all of our abdominal organs in place. Our skeletal muscle is considered to be an organ because it has the 4 tissue types. We have muscle tissue, connective tissue that surround fibers. We have blood and blood vessels with our epithelial tissue to help with circulation of nutrients, as well as nerves with our nervous tissue, and that help to lead to stimuli for contraction and relaxation of muscles. An individual muscle fiber runs the entire length of the muscle. However, it takes groupings of many muscle fibers to form the width of a muscle. So if we look at the gross anatomy of skeletal muscle, beginning from largest to smallest in size, we start at the muscle. The muscle is made up of multiple fascicles housing many muscle fibers. Here we find connective tissue coverings, we find blood vessels, as well as our nerve fibers. Then, going into a fascicle, this is a bundle of muscle fibers that are separated from other bundles of fibers by dense, irregular connective tissue. Now going smaller, we look at the muscle fiber, and this is elongated, multinucleated cylindrical fibers or cells, and contains myofibrils that are separated from other fibers by delicate layers of areolar connective tissue. And this is where we find striations of our skeletal muscle. Now going into myofibrils, these are long cylindrical contractile elements within the muscle fibers. And these tend to be as long as the muscle fiber itself. Here we find myofilaments, and these also exhibit striations that we see. And the striations are going to be divided by their unit of the sarcomere, where these are going to be separated by the Z lines. Finally, the smallest is the myofilaments of skeletal muscle, and these are short contractile proteins, and there are 2 main types. We have thick and thin myofilaments. Our thick myofilament is composed of the protein myosin, and our thin myofilament is going to be composed of the protein actin, as well as tropomyosin and troponin that help to hold the actin together and aid in contraction. The connective tissue of skeletal muscle includes the endomysium, and this is the innermost connective tissue layer. The endomysium is a delicate areolar connective tissue layer that surrounds and electrically insulates each muscle fiber. It also has reticular fibers to help bind together neighboring muscle fibers and support capillaries near these fibers. If you recall, the reticular fibers have a mesh-like, like a chicken wire organization, and so that really helps to stabilize and bind the surrounding capillaries to the muscle fibers. The perimysium surrounds the fascicles, And this is composed of dense, irregular connective tissue that contains extensive arrays of blood vessels and nerves called the neurovascular bundles. And these supply each individual fascicle with blood vessels and with nerves. The epimysium is a layer of dense, irregular connective tissue that surrounds the whole skeletal muscle. And if you recall, epi means upon, and so it would surround the largest of the muscles, so being the skeletal muscle itself. Fascia are going to ensheath the three types of connective tissue layers that we find within skeletal muscle. We have two main types of fascia. We have a deep fascia and a superficial fascia, and both are composed of sheets of fibrous tissue. For our superficial fascia, that is composed of areolar connective tissue, whereas our deep fascia, that is composed of dense, irregular connective tissue. The deep fascia separates individual muscles, and it helps to bind muscles together as well. And the superficial fascia helps to separate the skin from the muscles and helps to connect them as well. Fascia are also helpful in distributing nerves, blood vessels, and lymphatic vessels, as well as helping to fill in spaces between muscles. Plantar fasciitis is the most common cause of heel pain, where some of the symptoms include pain and inflammation of the plantar fascia, which the plantar fascia is responsible for connecting the heel to the toes. And so this is very common among runners, where a lot of pain is felt early on in the day. However, as things loosen up, the pain seems to go away by the middle to the end of the day. Tendons are extensions of the epimysium that connect bone to muscle. And the tendon is composed of connective tissue, and it can also connect muscle to skin or muscle to other muscles. And they tend to have a thick cord-like structure. Aponeuroses are flat, broad tendons that connect muscles to other muscles or muscles to other structures such as bones. And so we tend to find aponeuroses. One great example is within our skull on the very top, where we have the largest aponeuroses that connects the frontalis muscle to the occipitalis muscle. To reiterate the macroscopic anatomy of skeletal muscle from largest to smallest structure, we begin with our muscle, then we go to individual fascicles that are composed of muscle fibers. Muscle fibers are composed of muscle fibrils that are broken down into individual units of the muscle, which are sarcomeres, which are composed of myofilaments. And so fascicles, again, are bundles of fibers, and together fascicles will make up a skeletal muscle. The fibers are going to be long cylindrical cells, and these are different, though, from connective tissue fibers. And so fibers are also multinucleated, where they're found within the periphery of the muscle fiber itself. Myofibrils will be long rods of segments called sarcomeres that are separated by Z-lines. And so a sarcomere is a unit of muscle. And the myofibrils are composed of myofilaments, which are actin and myosin, are thin, which is the actin protein, and are thick, which is the myosin protein. And so we have our thin and our thick myofilaments. So again, with our skeletal muscle for macroscopic anatomy, from large to small, we have our muscle, and so these are composed of multiple fascicles, which will have the epimysium covering the muscle. Then we have our fascicles, which are going to be composed of muscle fibers, and these will be surrounded by the perimysium. And then we have the muscle fibers, and these are going to be surrounded by the endomysium. The myofibrils then are composed of sarcomeres, and sarcomeres are composed of our myofilaments, which again are our actin and our myosin. To look at the microanatomy of skeletal muscle, we'll begin at the level of the sarcomere. And the sarcomere is the basic unit of contraction of a muscle that is composed of thick and thin filaments. Our thin filaments are going to be composed of two strands of actin that are twisted around each other to form a helical shape. And so this has a resemblance of a string of beads. There are also two regulatory proteins that are found with the actin filaments. We have tropomyosin and troponin. The tropomyosin is a short, thin, twisted filament that covers a small section of the actin strand. And the troponin are going to be proteins that are found embedded throughout the thin filament, where it serves three functions. First, to attach actin to anchor itself in place. Secondly, to attach tropomyosin to hold it in place over the surface of the actin. And thirdly, for troponin, it provides a binding site for calcium ions so that contraction can occur. Our thick filaments are composed of myosin, and they are twice as large as our actin filaments in terms of diameter. Myosin has two strands that make up the thick filament, where each strand has a free globular head and an attached elongated tail. So think of it as looking like a golf club. The head would be the actual club part, and then the tail would be the shaft of the golf club. The long tails of the thick filaments point toward the center of the filament, and the heads point toward the edges and project outward toward the thin filament, so that during contraction, the myosin heads form a cross bridge by binding thick actin to thin myosin filament. Each sarcomere has a striated appearance to it, and these are due to different regions of the sarcomere itself. And so the Z lines are going to be the thin transverse protein structures that are going to be the boundaries of the sarcomeres. And so these are going to be in the center of the I bands that serve as attachment sites for the thin actin filaments. The A bands are going to be the length of the myosin filament, and they tend to have a dark appearance under the microscope. The H zone or H band is the center of each A band where there are no actin filaments. And so at maximal contraction, the thin filaments are pulled into the H zone, and the H zone disappears. The M line is going to be the center of the H zone where we find myosin filaments that are held together. And it keeps the thick filaments aligned during contraction and relaxation. The I bands are the light bands that contain thin filaments only, and so they look light under the microscope. And these are bisected by the Z lines. The sliding filament theory states that when a muscle contracts, the thick and thin filaments slide past each other, and the sarcomere shortens. So during contraction, we have the width of the A band remains constant. However, the H zone disappears. The Z disc in one sarcomere moves closer to another. The sarcomere narrows or shortens in length, and the eye band narrows or shortens in length. And so the thick and thin filaments, however, remain their same length whether a muscle is relaxed or contracted. It's just the sarcomere is what has changed. Motor neuron activity is what stimulates skeletal muscle contraction. And muscle contraction begins when we have a nerve impulse that stimulates an impulse to the muscle fiber itself. So each muscle fiber is controlled by one motor neuron. Now this is not the same as one muscle being controlled by one motor neuron. It's just a muscle fiber that is controlled by one motor neuron. The motor neuron transmits the nerve impulse to the muscle fiber at the neuromuscular junction, which is the point where the motor neuron meets the skeletal muscle fiber. The components of the neuromuscular junction include the synaptic knob of the neuron, which is an expanded tip of the axon. And here it expands further to cover a relatively large surface area of the sarcolemma of the muscle fiber, which the sarcolemma is just the plasma membrane of the muscle fiber. And so nerve impulses travel from the axon down to the synaptic knobs. Each synaptic knob then has synaptic vesicles, which are small membrane-bound sacs within the cytoplasm of the synaptic knob that are filled with molecules of acetylcholine, which is the main neurotransmitter for skeletal muscle. The motor end plate is going to be the specialized region of the sarcolemma, again, the plasma membrane of the muscle fiber, and it has folds and indentations to help increase surface area of the membrane that's covered by the synaptic knob, and so that we can have a larger recruitment of those larger muscle fibers. The synaptic cleft is going to be a narrow space that separates the synaptic knob from the motor end plate. Acetylcholine receptors are found at the motor end plate that act like doors that normally are closed, but will open when acetylcholine is present, when it's released from the synaptic vesicles of the synaptic knob. And then acetylcholine esterase is going to be the enzyme that is found within the synaptic cleft, and it rapidly breaks down acetylcholine so that we don't have continuous stimulation of muscles. Now, when we have continuous stimulation, this is what we find with tetanus and summation. And so, like when you have a Charlie horse, you have summation of increased reaction, and so we don't have enough acetylcholine esterase to break down the amount of acetylcholine that's found that binds to the receptors. With muscle contractions, what occurs is that first we have a nerve impulse that enters into the synaptic knob and travels down, opening calcium channels, allowing for extracellular calcium to enter in. This then pushes the synaptic vesicles down towards the ends of the synaptic knobs so that the acetylcholine can be released at the neuromuscular junction. Acetylcholine then binds to the receptors on the motor end plate at the synaptic cleft, which then initiates a muscle impulse. The muscle impulse spreads quickly along the sarcolemma and into the muscle fibers, causing calcium ions to be released into the sarcoplasm. The calcium ions bind to troponin, causing tropomyosin to move and exposing active sites on actin. Myosin heads attach to the actin, and then they form the cross bridges. The myosin heads go through a cyclic attach, pivot, detach, return as the thin filaments are pulled past the thick filaments. ATP is required for this detachment of the myosin head and completes the sequence of the cyclic event. The sarcomere shorten and the muscle contracts. This continues as long as calcium ions remain bound to the troponin. Calcium ions then are moved back into the sarcoplasmic reticulum by ATP-driven ion pumps to reduce calcium concentrations at the sarcoplasm so that we can have relaxation instead of a continuous contraction state of our muscles. The end of the muscle impulse results in the passive sliding of the myofilaments back to their original resting position. Motor unit is composed of a single motor neuron and all of the muscle fibers it controls. So motor units typically control only some of the muscle fibers in an entire muscle. Most muscles have many motor units, which means that many motor neurons are needed to supply the entire muscle. For instance, a large muscle like the gastrocnemius or the biceps has to have many motor units for contraction of the whole muscle itself. There is an inverse relationship between the size of the motor unit and the degree of control. The smaller the motor unit, the finer the control. So for instance, within our eyes, that help to move the muscles of the eye, we find smaller motor units but more precision in that control versus a larger motor unit within, say, our quadriceps muscle. However, it's less precise movements. All muscle fibers have this all or none principle where there's muscle fiber that is either contracts completely or it does not contract at all. So when a motor unit is stimulated, all of the fibers contract at the same time. So here we have an example of the axon of one motor neuron can have several synaptic knobs where they innervate several muscle fibers. And so when there is a stimulus from that motor neuron, in this picture, it would stimulate at least those 3 muscle fibers that that motor neuron is attached to. And so if we look at the actual junction between the synaptic knob of the motor neuron and the muscle, remember that term is the neuromuscular junction of where the neuron connects to the muscle. When describing the tension produced by skeletal muscle, this is also referred to as muscle tone. And so we have 2 main types of contraction that can influence muscle contraction, or muscle tone. So the first is isometric contraction, and this is where the length of the muscle does not change because the tension produced by the contracting muscle never exceeds the load. So for instance, if you're trying to pick up the back of the car, the tension that's generated is not great enough to move the load. However, the second type, isotonic contraction, this is where the tension produced equals or is greater than the resistance. And then the muscle fibers shorten, resulting in some type of movement. And so an example of this is when a person lifts a grocery bag out of the car, the tension generated in the appropriate muscle equals the load, and the muscle fibers shorten. There are 3 classes of skeletal muscle fibers, and however, most of our muscles contain a mixture of all fiber types, where we have one fiber type, though, that is predominant. The first fiber type are slow fibers, and these are red, and they're also considered our slow twitch fibers. We have our fast twitch fibers, which tend to be white in color, and they're also considered our fast twitch fibers. And we have intermediate fibers, which these tend to have like a pinkish color, and they are considered to be fast twitch as well. With our slow fibers, again, or our slow oxidative fibers, these are also known as type 1 fibers. They tend to use ATP slowly, and they make ATP in an aerobic fashion. There's an extensive amount of capillaries, and so another reason why we have that red appearance. The fibers, again, are red in color. They have a slow contractile velocity, They are the highest in resistance to fatigue. The distribution of our slow twitch fibers are found greatest in the muscles of the trunk, and especially with our postural muscles. The diameter tends to be the smallest and the thinnest. There are lots of mitochondria, and as well as lots of myoglobin, and the primary function is for endurance, such as marathon running, as well as for maintaining posture. Thus, these fibers are not considered to be very powerful and and aid in big bursts of energy. Our fast twitch fibers are also known as our type 2B fibers or our fast anaerobic fibers. So these fibers tend to use ATP very quickly, and they create ATP in an aerobic capacity. There are sparse concentrations of capillaries that are found throughout the muscle. We find that there is a white, pale color with these fibers. There is a high contraction velocity that is very fast, and it has then a low resistance to fatigue. The distribution of these fibers are found greatest in the upper limbs. The diameter of these fibers are the largest. However, they have the fewest mitochondria and the smallest amount of myoglobin. The primary function for these fibers is for short durations of exercise and intense movements. So sprinting and weightlifting are good examples because they need high and rapid bursts of energy. Our intermediate fibers are also known as our type 2A fibers and also known as our fast aerobic fibers. So these tend to use ATP very quickly. However, unlike the fast twitch fibers, the intermediate twitch fibers has the ability to make ATP in an aerobic capacity. There is an extensive amount of capillaries, and thus it's a little bit more red or pink in color. There is a fast contraction velocity, and it has a high resistance to fatigue, however not as high as our slow twitch fibers. We find intermediate twitch fibers in greatest abundance in the muscles of the lower limbs. The fiber diameter is intermediate compared to the slow and fast twitch fibers. There are lots of mitochondria and large amounts of myoglobin. And the primary fiber function for the intermediate fibers is for medium durations and moderate levels of movement and activity. And so good examples for this are when we do biking and walking. Cardiac muscle comprises the heart wall or the myocardium. The cells are individual muscle cells that are arranged in thick bundles within the heart wall. And these cells are also striated just like skeletal muscle fibers, but are shorter and thicker and have only one or two nuclei. The cells form a Y-shaped branch that joins to adjacent muscle cells at junctions called intercalcated discs. This allows for an autorhythmic activity where we have continuous contraction of the heart muscle that is in unison. It's also stimulated by the nervous system. The space that is found between the fibers is the endomysium, and cardiac cells also contain sarcomeres, but their myofibrils branch extensively, and so these are attached to one another by the intercalcated discs. Smooth muscle is found at the walls of visceral organs, within arteries, veins, and another one, the erector pili, which is found within our integument system. Smooth muscle is composed of short muscle cells that have a fusiform shape that are widest at the middle of the cell and taper off at each end. They have a single centrally located nucleus. Smooth muscle has both thick and thin filaments, but they're not precisely aligned, thus there are no visible striations or sarcomeres that are present. Z-discs are also absent from smooth muscle cells. Due to the lack of the Z-disc, there are dense bodies that are found, and these are where the thin filaments are attached by elements of the cytoskeleton. These dense bodies are small concentrations of proteins that are scattered throughout the sarcoplasm and on the interface of the sarcolemma. Smooth muscle is usually organized into two different sheet layers. We have a longitudinal layer and a circular layer. The longitudinal layer is primarily responsible for the strength of the visceral structure, whereas the circular layer is responsible for the expansion and contraction of that muscle. The contraction within smooth muscle is slow, it's resistant to fatigue, and also is sustained for extended periods of time. And so this is very important because for something like the blood vessels, we need to be able to sustain an extended contraction without undergoing fatigue. Smooth muscle cells are resistant to fatigue because their ATP requirements are significantly decreased compared to the needs by that of skeletal muscle. Most skeletal muscles extend between bones and cross at least one mobile joint, so upon contraction, one of the bones moves while the other bone usually remains fixed. So the less mobile attachment of a muscle is the origin, and the more mobile attachment of the muscle is its insertion. Usually the insertion is pulled toward the origin. In the limb, the origin typically lies proximal to the insertion. So for example, the biceps brachii originates on the scapula and inserts on the radius. The contraction of this muscle pulls the forearm toward the shoulder. Sometimes the insertion and origin cannot easily be determined, and so different criteria are used. So for example, the muscle that extends between the broad aponeuroses and a narrow tendon, the aponeuroses, is considered the origin, and the tendon is attached to the insertion. Skeletal muscles generally do not function in isolation, rather they work together to produce movement. So we have agonists or prime movers, and these are muscles when they contract to produce a particular movement, such as extending the forearm. So the triceps brachii of the posterior arm is the agonist that causes forearm extension. Synergists are muscles that assist the agonist in performing their action. The contraction usually either contributes to tension exerted close to the insertion of the muscle or stabilizes the point of origin. Synergists are most useful at the start of a movement when the agonist is stretched and cannot exert much power. For example, we have the biceps brachii and the brachialis muscles of the arm. These would be considered synergistic muscles because together they help to flex the elbow joint. Antagonists are muscles whose actions oppose those of the agonist. If the agonist produces extension, the antagonist produces flexion. The contraction of the agonist stretches the antagonist and vice versa. So for example, when the triceps brachii act as the agonist to extend the forearm, the biceps brachii on the anterior side of the humerus acts as an antagonist to stabilize the movement and produce the opposing action, which is flexion of the forearm. With skeletal muscle fiber organization, we have circular muscles, and so these are concentrically arranged around an opening or a recess. A circular muscle is also called a sphincter because contraction of the muscle closes off the opening. So these are located at entrances and exits of internal passageways, and so one example is the auricularis oris, which is the muscle that encircles the opening of the mouth. Convergent muscles are widespread muscle fibers that converge on a common attachment site, where this attachment site may be a single tendon, a tendinous sheet, or a slender band of collagen fibers known as raphes. These are often triangular in shape, and they resemble a broad fan with a tendon at the tip. This is a very versatile muscle because it is pulled in all directions, and thus it can modify by activating a single group of muscle fibers at any one time. And so one example of this is the pectoralis major of the chest. Parallel muscles run parallel along the long axis. Each muscle fiber in this type of skeletal muscle has the functional characteristics of the entire parallel muscle. The parallel muscle has a central body called the belly or a gaster. When these muscles shorten with contraction, the body increases in diameter. Thus, these muscles have high endurance but are not as strong as other muscle types. Examples of these are the rectus abdominis, which are responsible for your six-pack, the biceps brachii of the arm, and the masseter, which is responsible for moving the mandible. Pennate muscles have their tendons and muscle fibers that resemble a large feather. They have one or more tendons extending through their body, and the fascicles are arranged in an oblique angle to the tendon. When the muscle fibers pull at an angle to the tendons, this does not move its tendons as far as parallel muscles move their tendons. Because of this too, though, this generates more tension than a parallel muscle of the same size and is stronger than a parallel muscle. There are three types of pennate muscles. The first is a unipennate muscle, and this is where all of the muscle fibers are on the same side of the tendon. An example of this is the extensor digitorum, which is a long muscle that extends the fingers. A bipennate muscle. This is the most common type of the pennate muscles, and it has muscle fibers on both sides of the tendon. The rectus femoris is an example of a bipennate muscle. And then the third one, a multipennate muscle, has branches of the tendon within the muscle. So the triangular deltoid that covers the superior surface of the shoulder joint is an example of a multipennate muscle. Muscular dystrophy is a term for several hereditary diseases in which the skeletal muscles degenerate, lose strength, and are gradually replaced by fat and fibrous connective tissue. The new connective tissue impedes blood circulation, which further accelerates muscle degeneration. The most common forms are Duchenne muscular dystrophy and Becker's muscular dystrophy. These are groups of congenital diseases that are predominantly affecting males, where 1 in about 3,500 live births occur within males. These individuals have muscular difficulties that become apparent early in childhood, where walking is a problem, the child falls frequently, and has difficulty standing up. The hips are first affected, followed by the lower limbs, and then eventually the abdominal and spinal muscles. This muscular atrophy causes shortening of the muscles, which results in postural abnormalities like scoliosis. And usually these patients are confined to a wheelchair by adolescence. Unfortunately, individuals rarely live beyond the age of 20, and currently there is no cure yet today. However, some studies have found that electrical stimulation and physical stimulation, like with exercise, has been shown to have beneficial effects, where it does help to slow down the rate of the connective tissue and adipose tissue that takes over the muscular tissue.